People will start coming in now. So those of you who are joining us now, this is Superhuman Radio. We're officially 20 years, the longest podcast for health, fitness, and anti-aging. And people are starting to join us now. And today we're going to be talking to Irina Conboy, and she is someone that I have fallen in love with her brain. She's been on the show actually either 3 or 4 times, once with your husband when you're over at the Conboy lab. Mm-hmm. And now you're at Generations, the lab that you're at, right?
That's correct.
So I learned a lot from you. You know, we talked about plasmapheresis. The first interview I ever did with you, we talked about oxytocin and how it makes old muscles young again.
Yes, that was back in 2014.
Yeah,
more than 10 years ago. Yes,
I know. And I'm actually leveraging that research because I had sepsis and was reinfected in January and February, and I ended up having spine surgery because all of the discs in my spine got infected. And I'm leveraging a drug that increases oxytocin because I'm hoping that'll help my old body bounce back. But yes, I've learned so much. I could be wrong, but I have quoted you as saying something once on a show that really changed the way I look at anti-aging. I think it was you. Correct me if I'm wrong. During one of your interviews with me, you said to truly have effective anti-aging results, it's not about what you take; it's about what you remove from the plasma.
Well, kind of, right? What I probably said is that aging is not decline in things; it is an excess in various circulatory proteins that, when they are present at healthy young levels, are really vital for us. But then when they become age elevated, then their functions change in ways that are counterproductive for health of our many tissues in our body. So yes, so if you think about, should we add more vitamins or should we calibrate systemic determinants of our tissues back to youth and diminish them? Yes, that's correct. We should probably diminish them as the first step in the right direction.
And I've started to think about aging as the accumulation of metabolic debris in the body, in the cells, in the blood. And that's why I tend to, I can't do it now. I haven't been able to do it because I was anemic after I got out of the hospital. But I believe in donating blood because you get rid of plasma, which we learned from one of your interviews. There is misfolded proteins accumulate, and that's why the value of plasmapheresis was proven by your research. And so it's almost like you change the oil in your car, but you don't change the oil in your body. You don't do anything. It just circulates from the day you're born to the day you die, and it just gets dirty.
Right. So on the topic of plasma donations, my husband, Michael Conboy, who now runs the Conboy lab at University of California, Berkeley, donated his plasma today, which was his second donation. And yes, and to follow up on your introduction, I am Chief Science Officer at Generation Lab, which is a spin-off Conboy lab. So I spent over 20 years discovering things in the sphere of aging and longevity. And now it is time for me to switch gears, to shift gears, to pursue their clinical translation. We have discovered enough, in my opinion, to start thinking of rational approaches for rejuvenation of multiple tissues. Moreover, in people who are already old and frail, starting with people who are already old and frail. So your analogy with changing oil in the car is good. Another analogy is changing water in your fish tank. So if you just have water there and there is accumulation of debris and whatever fish poops into the water and the plants, then eventually the whole fish tank ecological system becomes unhealthy.
Mhm.
But if you change water too frequently, you also kill all of the fish in plants. So we don't have kind of one-off solution that you just keep donating your plasma or keep doing therapeutic plasma exchange. That was just an indication of the direction. Do we get younger by adding things to old systemic milieu, or do we get younger by removing things? So that was kind of a fork in the scientific road.
So let's do something and just kind of do a cursory review of some of your studies that I feel are the most impactful. So
Yeah.
let's talk about the first one about the old rodents and the young rodents and oxytocin.
Oh, that no, was not the first one. That was like 10 years after I started my position at UC Berkeley. But yes, for us, for our discussion, it probably was the first one. Mm-hmm.
And when you took the old rodents and you,
Mice.
or they were mice, I'm sorry,
They're mice. Uh-huh. They're
old,
rodents, mice are rodents, but just to be more specific, old mice.
yeah, and you inflicted an injury in the basically the quadricep muscle,
Not
and
quadricep, below the knee,
okay,
tibialis anterior. Mm-hmm.
okay, and then and compared the recovery of the old mice and the young mice. The young mice recovered faster naturally,
Mm-hmm.
but when you applied oxytocin, infused oxytocin to the old mice, they actually recovered as fast, if not faster, than the young mice, right?
Not 100%, right? So they improved significantly as compared to control old mice that were injected with just like vehicle, let's say saline. But it was not is the same as young mice. And by the way, that was one of the few pharmacological approaches that we pursued for rejuvenation. The first one started back in 2003 when Mike Conboy and myself were postdocs in Tom Rando laboratory. So we actually activated one of other signaling molecules called Notch in old mice. It is published in Science in 2003, and that also produced rejuvenation of skeletal muscle. But in none of these experiments, old muscle repaired as nicely as young. It was repaired much better than old, but not as nicely as young.
So there is a drug I'm using for my recovery, and it's an anabolic steroid. And I'm not suggesting people do this, but it's an anabolic steroid called trenbolone. It's used on bovine, on cattle,
Mm-hmm.
and it has the ability to make uh, cows to add a pound of muscle a day.
Wow.
Yeah, and the way it does it, and this is going to fascinate you. I
Yes.
was looking to be able to talk to you about this. It raises serum oxytocin levels 50-fold, 50,
Yes.
50-fold. And it's the strongest anabolic steroid out there. Its
Mm-hmm.
pathway is that it raises oxytocin. So I'm just taking a minuscule micro dose, 10 milligrams
Mm-hmm.
a day when I take my testosterone. And I can see I'm getting stronger faster. My doctor has said it. My muscles are coming back because I lost 75 pounds from the sepsis. And
Mm-hmm.
thank God I had muscle to lose, or I would have died in the hospital. But I,
Wow.
I really think that trenbolone should be viewed for older, aging men because it's androgenic, so you can't give it to
Mm-hmm.
them. And also because it raises um, oxytocin levels so much, it makes women with the uterus they get cramps because the uterus is contracting because of the oxytocin.
Yeah.
So
Wow.
yeah, but it's I really think doctors should investigate trenbolone in small amounts because it'll make old muscle like young, you know?
Well, I don't know if it will make old muscle like young. You are completely correct, Carl, that everything that we hypothesize to happen should be researched first to
Right.
know not only efficacy but most importantly safety. Trembolone binds to androgen receptors and also glucocorticoid receptors in addition to
Hmm.
many other pleiotropic functions. So it's great to use it how it is prescribed to use. I would not recommend right away to switch the direction of how it is used
Right.
without doing efficacy and most importantly safety.
And I agree. I'm being reckless, but I'm desperate to recover from the disaster. I like to say
Mhm.
my body has become a human shipwreck
So
between
no,
the spine surgery and the muscle loss and the sepsis. Uh,
yes.
I don't sleep well anymore. I mean, it's all a result of the sepsis. So that's why
Hmm.
I'm just using a little 10 milligram shot, and
I would just say like talk to your primary care. I am not a physician, so I cannot really
no, no,
meaningfully or
I know,
recommend
I know
anything.
that, and I'm making these statements, not you.
Yes.
But one of the things that I want to talk about is parabiosis, which has become
Mm-hmm.
very popular, and this is when you take the blood from young people and you give it to older people, right? Do you have to when you
So
do
parabiosis is an ancient procedure, like 200 years old, where two animals are physically, surgically sutured to each other. It
oh,
obviously cannot be used in clinic or in people. So it is just pure experimental research where, like we in 2005, we published paper on two mice, young and old, sutured with each other, and then young mouse became older, old mouse became younger. But
that's
but
amazing. Well, I want to recover. I don't want
yeah,
that's an amazing thing. The young mice became like old mice.
not
And
like old, but they became rapidly aged.
they
Not completely
rapidly age.
100% like old. And then even more in a better way, we just exchange blood between young and old mice. And when young mice, with all of their like perfect telomere length, good mitochondria, good joints, muscle, everything is young, and they still have 50% of young blood. They receive 50% of old blood. In just 2 weeks, they become old and senescent in muscle, in liver, in brain, in kidney, in their blood circulation. So aging that we perceive as just the progression of time is a regulated process. We can inflict aging upon experimental animals by 50% of old blood in just 2 weeks before there was any time for them to age naturally.
That's fascinating.
Hmm.
And a lot of your discoveries are with the ability for blood to affect uh, biological age. I mean, it's fascinating.
Hmm.
So when we were on last time with you and Mike,
Hmm.
we talked about uh, removing. So we talked about
Right.
one. We talked about removing plasma and putting synthetic plasma in, which is basically saline and some
Albumin.
vitamins, albumin,
Hmm.
and that's actually being done in clinics right now, and it's not cheap. And
Absolutely.
I have people tell me that they did two in one weekend, and they felt dramatically different leaving
Yes.
like in days. So
Well,
is it realistic to expect to feel different if you're doing plasmapheresis, where removing and replacing? Is that a common trait that people feel that good that quickly?
I don't think I cannot tell that if they felt good. They definitely would feel different because 70% of your blood was drained from your veins. And the reason that people are not dying is that simultaneously with that, in the second IV, they get infusion of saline and albumin. And then their cells are being either filtered or spun down and returned back to them, the blood cells, like red
Right.
blood cells and then white blood cells that fight infections. So everybody would feel woozy, lightheaded, or kind of different from this procedure. With respect to rejuvenation, we published, I think, a seminal paper in 2022 saying that if you look then at what happens to blood cells 1 month after procedure, they become less damaged in their DNA
Hmm.
and less senescent. So there are long lasting positive effects. And furthermore, there are less inflammatory cells and more productive immune cells that can combat viruses, cancers, and infections. And that happens in people who already are older. And furthermore, many of the systemic determinants, those proteins circulating in our blood, become also younger and healthier. So we published that paper. And it was on people who underwent several consecutive rounds of procedure, not just one, but every month or so.
Right.
And but then another observation was that after a couple of rounds, the effects become taper down or plateau, and we do not know if there is any additional benefit. So it is an immediate improvement, but because your organs and systems continue to be old and damaged, they will still be cranking up the production of extra proteins and sending them into your bloodstream. So then, to become meaningfully rejuvenated, the person would then need to do multiple procedures all the time.
Mhm.
And because the procedure is not completely benign, right? That is invasive kind of procedure where your blood is drained and cells are spun down and put back into you. Then my fear is that the side effects will start overpowering any beneficial effects if you keep doing this plasma dilution constantly, trying to offset the damage to your body.
Right. And plasma dilution, the plasma replenishes what in 2 days to 3 days.
No, it's about, I would say, a couple of weeks. So
Okay.
some things will replenish right away. Um, by the way, this procedure will have certain, load on your cardiovascular system because bunch of liquids are removed and then put back, as well as on kidney and liver because the cells from your blood are being filtered and then put back. Some of them are damaged by this procedure, and liver
Right.
has to deal with that, and kidney, right?
And
And
that
then,
was the thing I was afraid of because of plasmapheresis because they spin the red blood cells
yes.
at I think 20,000 RPM, and I would imagine if they're damaged when they put them back in you.
Well, they know what they're doing, right? The procedure was in clinic for 35 years to treat deadly diseases. And
Right.
so, if you are in need of this procedure, for example, people who OD'd, the best way to rescue them is to do plasmapheresis, to remove all of the drugs from the system. Or people who have autoimmune diseases, they have antibodies that attack them in blood. Same thing. So if you are doing it for clinical reasons, because otherwise your disease progress, absolutely. And clinicians know how to do it. If you are doing so you can play tennis or golf better and you are otherwise healthy, I would caution to talk to your primary care because multiple rounds of procedures would be needed constantly. And I don't know if there was any study to say that if you do it like for a year every month, it is safe.
I remember probably 12, 13 years ago reading a study that indicated that donating whole blood 2 to 3 times a year
Mm-hmm.
seems to improve longevity and improve
Absolutely.
disease things. It does, huh?
Absolutely. Right. Absolutely. So blood donation, right, are much milder because it's not like most of your blood is removed. It's like one
Hmm.
unit, right? So that is milder, and the process is exactly the same. So the plasma is discarded, cells are returned back to you in saline, and you don't need probably to add albumin. They can add albumin, but your liver produces albumin by itself. So and then it is done all over the world. So it would have beneficial effects, but I would caution people to consider it the solution for rejuvenation for people who are relatively healthy.
Hmm. I remember Mike laughed at me because I was interested if consuming chicken albumin, egg albumin
Oh
was
yes, it's
beneficial.
not
He
the same.
laughed at
No,
me. I
it's not the
said, well,
albumin.
maybe it's good for you because I
No.
know there's a couple biological age tests. I'm trying to think of the doctor who created one, very popular, but they look at your blood work and they look at the amount of albumin as very beneficial and positive. The more.
So what? Yes, what you should know is that we looked into that in our publications, And albumin is the key, it's most abundant protein in our blood that we produce in the liver. And it is simply needed to keep blood vessel rheology or diameter intact and for many other kind of structural and key biochemical functions. So albumin does not decline significantly
Right.
between people who are young and people who are old. And there are, controversial observations, but sometimes people say it declines maybe by 5% when you are 75 as compared to when you are 25. But other papers say that no, it only happens when people are also very sick and their liver doesn't work. So I would not say that restoration of albumin is the main function of plasmapheresis.
Really?
Exactly. I think the main function is physical removal of age elevated proteins that are overproduced in the cells of your body. If they are age elevated, then the signaling that the instructions that they send to cells become perturbed. They need to be present at certain levels in order for the cell-cell communications to be accurate.
Yeah, and I guess that a lot of disease states seem to include damaged protein in
Yes,
well,
absolutely. So there are many diseases of protein misfolding, protein aggregation. And in the paper that we published in 2024, we show that it could be easily explained if you look at the rate of production of new proteins in cells, young cells versus old cells. Old cells overproduce proteins
mm,
globally. So many proteins become now overexpressed, overproduced. And furthermore, not steadily, but again, fluctuating for high to low. And that poses like a lot of challenge for the cell quality check or control check to make sure that only healthy and properly folded proteins make it where they should be. So absolutely, we believe that there is a link between protein overproduction and diseases of protein aggregation and misfolding, for example, Alzheimer's disease.
is amyloid a protein?
Yes,
Okay, so I'm going to be in.
amyloid, there is like amyloid precursor, right? APP precursor protein, and then there are beta amyloid isoforms, which are
Right.
cleaved parts of that. Hmm.
So Anthony Samsel, who is one of the think tanks on the East Coast, I want to say Arthur Daniel
Hmm.
something think tank. He came on my show quite a while ago. This is the beauty of being on the air for 20 years. Uh,
Yes.
yeah, and the new science comes out, and so he's coming back on the show next month because he talks about glyphosate exposure causing a phenomenon called amyloidosis.
Hmm.
Amyloidosis, he feels, is at the root of the uh, over. My brain doesn't work as good as it used to.
No, no, no,
So
I know what you mean. Probably, the appearance of pathogenic amyloid beta peptides.
yeah, and so he says that it's coming from exposure to glyphosate, the weed killer. He
Hmm.
said, and he just published a new paper on the how the exposure to glyphosate by the population
Hmm.
is higher than they think it is, and
Right.
it's leading to um, neurodegeneration and what is it? Uh, not
Neuroinflammation, right? Neurodegeneration, neuroinflammation. Hmm.
well. What is the common one that diabetic people get? Um, Peripheral neuropathy. He says it's at the root of the neuropathy. He said we have a population that's not even diabetic and they're all developing neuropathy. He says it's
I
because of
see.
the exposure to glyphosate. He's going to come on and talk about his paper next month.
Yeah, I don't know. Carl, in my opinion, we always try to catch like silver bullet solutions
Right.
or to find silver bullet solutions. Um, we have neurodegenerative diseases simply because now we live much longer. So biomedicine did marvels, for us with
Right.
antibiotics and also development of the civilization. So we have new better nutrition, we have sanitation. So people in the past simply did not live long enough to start developing cancer flares and
Right.
Alzheimer's disease and Parkinson's disease. So now we have the whole host of this diseases of later years of life, and I think we should rethink our approaches of how we then solve them and how we treat them. Because I would not say that it's only one molecule which causes Alzheimer's. And Alzheimer's persists in the environment where the whole body is old. Immune system is not working well. Microglia cells, which are immune system representative in the brain, do not work well.
Right.
So when we try to develop like a drug for Alzheimer's, we only think of a brain, but we should think about the entire body. Can we now rejuvenate various body systems so our drug actually has a chance to work? So that's kind of my take on that, that I'm not a fan of silver bullets.
If anybody has any questions, you can post them uh, on your uh, in your browser. There's a place to post comments and stuff if you want to. So I'm fascinated by the idea of donating blood.
Mm-hmm.
It's a very simple thing that can be done. I would imagine that that is something that we could tell people in general. They always need blood. Donate blood once or twice a year, and it could be an overall benefit to people.
Yes, but I could not tell them that it would be overall benefit. It certainly would be benefit to society unless
Yes.
we do the study. So for a while now, Mike Conboy and I and other people, for example, from gerontology research group run by Johnny Adams, were thinking that it would be great to have a clinical trials, simple clinical trials on people who donate blood, and we can do it longitudinally to see if there is any improvement in their biological age, which, by the way, Generation Lab, the company that is co-founded, quantifies, not predicts like, so-called biological clocks, which are just predictive models, but quantifies from intact primary data person's biological age accurately and reproducibly. So absolutely, we would be very interested to see which
Yeah.
approaches, right? What works, what doesn't work. Many people use stem cells or they use therapeutic plasmapheresis. How about plasma donation? Now, for the first time, we have a measure to tell you directly: is it working? Is it not working? If it's not working, why? Can we improve it?
When I was in California not too long ago, I went and had stem cells infused. Just what do they call it? A push. It was intravenous, and they assured me that these stem cells were not coming from people who received the COVID vaccine. And I see people talking about stem cells now pre-COVID. Like, so did the COVID virus itself cause lingering changes in the people that had it? Because I had COVID. I mean, I didn't get the vaccine, but I had COVID. What do we need to know about that sort of stuff, especially when we're doing blood work with blood infusion?
So I am a opponent of injecting somebody else's cells or body fluids in yourself. I say do not do it. By the way, stem cells die almost immediately after they've been injected. So I don't understand why there would be any benefit. That has been shown numerous times in experimental research. And it is expected because stem cells are not just, kind of magicians that can repair body wherever they find themselves. They need detailed instructions what to do. And if somebody's so-called stem cells have been isolated from a person's fat and injected to your IV, they will be just bewildered. Where am I? What am I supposed to do? And the response to that, as well as from the shear of going through syringe, is that they will die.
Um,
They will produce certain cytokines or molecules, right? Growth
right.
factor cytokines, but there's not enough of them, and it's only one single application. So personally, I'm not proponent of stem cell injections or anything that is biological product derived from somebody else to inject into a person. I don't know.
I didn't notice anything from the stem cell infusion. Nothing. I was trying to modify my pain because I have so much pain, but it's getting better. But I didn't
That's
notice
good.
any.
I'm glad.
But isn't donating blood doesn't that stimulate the production of your own stem cells?
Yes, under some circumstances, you know that. It does not stimulate their production that much as it will stimulate their mobilization. So these hematopoietic stem cells from bone marrow migrate into the bloodstream, and they produce more B cells there, B cells that make antibodies for you. But it is very, very limited in magnitude effect. And we don't have anything that we can inject into a person right now and make the person younger. Either it is a stem cell or it is exosome. So when people are using it, it makes me very nervous. Maybe because I'm not really, clear medical expert in the field, so I cannot give any medical advice to begin with. But
Right.
when I think about it from perspective of a researcher, it makes me nervous.
And exosomes are produced by stem cells, right? That's why they all
Exosomes are produced by every cell in the body.
right. Yeah, I know they're very popular. People are spending tens of thousands of dollars, and I really think a lot of it has to do with placebo.
Yes.
They believe, they want to believe. So I wanted to believe so much that it was going to make my back pain go away, but it didn't.
Sure. So what I suggest for people to do is to look at, famous researchers, famous scientists. Do they inject themselves with stem cells or exosomes? They published on that. They are the ones who, made breakthroughs and published seminal papers. Do they use it? If they don't use it, they are the ones who know the most. And the question is why? Why most of the famous scientists in the field are just doing diet and exercise and nothing else?
Right.
It's because, in my opinion, we don't have anything else. We can develop it. Generation Lab is, in fact, working on meaningful next generation longevity therapeutics. But right now, there are none, in my opinion, that would be better than good diet and exercise.
You also do testing, right? You do, what do they call it? Age, biological aging?
Biological age and risk of disease, that is the only product that Generation Lab has right now. And it is based on our discovery. In 2023, the Conboy lab discovered what is our fundamental curve of biological aging. How do we go from being young to becoming old? It's not obviously a straight line, right?
Right.
Everybody knows that you do not grow one gray hair per day, and that's how you have the head of gray hair. So it's not how you grow. So then there are alternative approach, which is just to have a computer driven model where you have young biomarkers, old biomarkers, and you artificially connect them in a straight line. The straight line does not exist in real world, right? You just made it on a computer.
Mm-hmm.
So that does not allow you to answer this key question: what is the real line? It's not straight line. What is it? So then in 2023 we discovered what that line is, what is the actual curve. And systems age done in due to the work of the systems age data scientist Rinting Han, then discovered that this curve is fundamentally the same for different functions of our body. For example, if you look at the aging of inflammatory system, immune system, it will be the same curve. If you look at the aging of cardiovascular system, it will be not exactly the same, but very similar pattern. And the same will be true for regeneration, fibrosis, reproduction, and so forth. So that is our 19th systems age, where from intact primary data without data transformations, we can tell you where are you on that curve of aging.
Right.
There is a healthy plateau, obviously, right? There is healthy plateau of adulthood. Are you on a healthy plateau? Are you deviating upwards towards increased age way too early for your calendar age? And then we also do the service for clinicians. We have 275 affiliated clinics where by using the AI on what is published on PubMed, we then tell clinicians that based on PubMed literature, this might be the best course of action for a person who has certain premature changes in biological age.
Mm-hmm.
So that's what Generation Lab product is.
It was the test that I was trying to think of before was the Horvath clock. Horvath.
There are many of them, right? So there is Horvath, there is
that's the one. That's the one that
Osage.
focuses on albumin. That one focuses on albumin.
No, 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 no. It focuses on DNA methylation.
Oh,
It can
methylation.
right DNA methylation in um, blood cells, peripheral blood cells, and there are many other ones. There is pheno-age, there is damage, cause age, pace of aging. So they're all variations on the same correlative
Yeah, but
approach.
I always I see these websites, these companies saying, there's a guy who's was a vegan. He's taking 100 pills a day, and if you follow his protocol, you live longer. And I went on his Instagram. I said, "How much longer will I live? Like, give me a
Mhm.
quantity. Can you tell me I'll live 10 years longer with if I follow your?"
Yes.
No one knows. No, nobody knows. They make these statements. It's silly.
Well, yes, it is. It is kind of silly, and also it is correlative, right? Correlation is not causation. So what distinguishes systems age of generation lab that it is causal and that it is not really reliant on any sort of arbitrary data transformation, which is biologically arbitrary and mathematically serve the purpose of making a line graph from young to old. So that what is the distinguishing characteristic. First, you need to have a hypothesis. What is the progression of human aging? And then you need to use data that is intact experimental data to figure it out.
So we have a person, Craig Hollander. He says hematopoietic stem cells can be stimulated through various methods, primarily focusing on mimicking their natural bone marrow niche or using this is a long
Mm-hmm.
pharmacological agents.
Yeah,
But
AGM. Okay.
but what
Yeah.
do they do for us? Like
Yeah.
what can I expect to happen if I follow these protocols and I stimulate these stem cells? You know?
So co-culture would require taking the cells out of your body, right? They live in your bone marrow, and you would need to isolate your bone marrow and then culture the stem cells
Mhm.
or activate them in some kind of way. But what was published in a very elegant study, again, I think it was 2014 or 2015, with a sleeping beauty transposon,
Mhm.
that it is not stem cells but more differentiated progenitor cells in the blood, that in the bone marrow, that keep constantly giving us blood lineages like red blood cells and various types of white blood cells. So stem cells are typically just sleeping, and then when something, maybe again it is just my guess, traumatic happens, like person all of a sudden has
Like
irradiation.
somebody gets sepsis twice. Yeah.
Exactly that. Then they wake up and they quickly repopulate the bone marrow and make more progenitors, then make you final blood lineages. But it is well known that stem cells age, and when they age, they are skewed towards making progenitors of myeloid lineage, such as inflammatory macrophages, and not enough T and B cells. And so there are many approaches, for example, retrobio, not approaches, but thoughts. Retrobio is thinking seriously on rejuvenating hematopoietic stem cells, which, in my opinion, is an interesting thought.
Interesting.
Mhm.
I think the most interesting thing that you talk about and is always about the manipulation of blood, and because it makes perfect sense. Uh,
Mhm.
it's like I said before, like everything gets dirty in the blood, and if we
No,
don't
no, no, no. I would not say everything that gets dirty in the blood. We have blood that
well,
serves
I'm
us
calling it
differently.
dirty. I'm saying it's metabolic debris. It's debris,
No,
stem
it is
cell.
not.
I mean,
It is not metabolic debris. So it is not a problem, right? So we have our kidney and liver, and the blood is filtered and kept in actually good shape throughout our life. So it is not that you would imagine that during your life you keep accumulating debris and now you have the whole garbage dump in your blood. No, you don't. You do not accumulate debris. But when you grow older, the cells all over the body becomes damaged, and when they are damaged, they change their gene expression. So they are no longer doing pristine, controlled use of the software that is in them. The software becomes somewhat corrupt, and so many of the genes become overproduced, and many of the proteins become overproduced. For example, transforming growth factor beta. We cannot live without it. We cannot even go through embryonic development without it. But in old people, it's elevated 3 to 6 times. And when it's elevated, it starts doing different things. So that is the problem. It's not that we simply are washing away metabolic debris. No, we are not doing it with plasmapheresis. We are bringing the levels of the proteins to younger levels, but then in a couple of weeks they go back up because cells
Yeah.
remain damaged. Yeah.
So you have to keep doing it.
You have to
Robert
keep doing it.
Robert Downey says Vesogen can stimulate stem cells. Is that a product? VESU, is that a RGEN? Vesogen? Have you ever heard of that?
I'm Googling it right now. Vivogen and stem cells. Synthetic tripeptide.
Oh,
Yeah. Particularly influencing stem cells and neurogenesis. There are many things that can stimulate stem cells, right? Potentially. But they live in the environment of the old niche. So all of the cells around them are telling them to do something else.
right.
And so then the question becomes how much vestrogen can one person deliver to brain if it is for neurogenesis, and is it safe otherwise?
It's interesting because we just did a show a couple days ago. There's a peptide that I'm using aggressively right now. It's called thymosin beta 4,
Mm-hmm.
but not TB500, because a lot of peptide companies sell TB500, which is just a portion of the
Yes.
thymosin beta. But thymosin beta 4 has been shown in rodents to switch on neonatal genes.
And why do you want to do that, Carl? Why do you
Well,
want to
because
switch on
I have a
neonatal
lot of
genes?
a lot of damage from the sepsis. My organs, my kidneys were messed up for a while. So I want my
Yes.
body to have the ability to turn on the machinery to repair this stuff.
Well, neonatal genes are not necessarily good for you to repair your stuff. And plus, there is not a single molecule which will preferentially switch on all of the neonatal genes all over your body. But what can happen is that they will have dysregulation. It will have some unusual genes and proteins that cells cannot recognize
Mm-hmm.
because they are supposed to be turned off. We switched from neonatal to adult program for a reason, right? And every single protein is looking for its partners with which to work to keep
Right.
cell healthy, right? So yeah, I would suggest that maybe we should think of returning to the healthy adulthood instead of to becoming an embryo or a neonate.
Right.
Like one, yeah, exactly. That probably would be more prudent to do. Healthy
Well,
adulthood.
I'm going to donate blood because I'm not. I was anemic after the sepsis because the antibiotic that I was getting for 6 months, it slowly lowered my red blood cells. So now
Mm-hmm.
I'm back to normal. So I should be able to donate blood.
Yeah, or plasma. Donate plasma.
Yeah, I could just donate plasma, right?
Mm-hmm.
I'm already signed up for the plasma pheresis clinic in my area, so I could just
Mm-hmm.
go there and donate plasma.
Yes. Octa Pharma.
Interesting. So what is on your website that my audience can take advantage of? Is there anything that they can go there and purchase?
Absolutely. So I'm chief science officer. I do not do any sort of like marketing. Alex Plant does that, or your business parts, which is
Mhm.
Alina Sue and Michael Saswell do. So by the way, Alina Sue is our CEO, and Michael Saswell is chief operating officer of Generation Lab, and both of them are successful, multiple multiple entrepreneurs. So they had successful companies before Generation Lab. So on our website there is a detailed explanation of how to use systems age, which will tell you where you are on the aging progression for various 19 different functional systems of your body and organ systems. And again, that is not
I need to
correlated.
do that. I
Yeah.
need to see what is working and what's not.
What is working, what is not, right? And particularly sometimes there is a weak link. So for example, various systems are okay, you are where you should be, or even younger by a couple of
I'd
years,
be
right?
surprised if I was where should be after this fiasco this year. But
but
maybe
but
you
maybe
know,
there is like one or two that are particularly getting old faster, and then we have, billions of published papers on PubMed engine, and Generation Lab has AI to tell, okay, out of all of these papers, which ones are reliably telling you that this might be the way to improve
Yeah.
things. And
Yeah.
then, and we do not really either, we do not sell supplements, we are not like what you mentioned, tell that buy the supplement from us and you will become younger. No, we don't do that. We are very agnostic, so we just have free of charge recommendations for clinicians who then can review our recommendations, agree or disagree, and then design the better treatment plan based on
Yeah.
those.
So that Generation Lab or Labs?
Lab, Generation Lab, like laboratory.
Labs.com.
.com.
So
Uh-huh.
people can go there and learn more. And
Yeah.
also, there's a list of published papers,
Mm-hmm.
fascinating stuff that your group has done. Some of that was done while you were at the Conboy lab. Yes.
Yeah, I have left Conboy Lab like less than 2 months ago. So so it was
Okay.
all done by the Conboy Lab.
Okay. Is there anything else you want to cover?
No, thank you so much, Carol, for
I
your
think this is good.
continuing
This is
interest.
fun.
Right?
And
Mm-hmm.
and Dr. Irina Conboy is a wealth of information about aging and anti-aging, but
Mm-hmm.
because she's published some great papers that I refer to all the time. So check out generationlab.com.
Mm-hmm.
We'll see you soon.
Bye.
We'll see everybody in a couple days. We'll be back on the air. Thank you.